Hello and welcome to week 4 of HAP 201. In this unit, we will be studying the heart. The heart valves are very important for directing blood flow. They open and close in response to pressure differences across the valves created by the different phases of the heart, be it contraction or relaxation. The contraction phase of the heart is called systole, and the relaxation phase is called diastole. Blood always flows from areas of high to low pressure, and it is the contraction of the heart chambers that increases and creates the pressure. So the opening and closing of the heart valves ensure one-way flow of blood. The atrioventricular valves are the tricuspid on the right and the bicuspid on the left. And again, these separate the atria from the ventricles. The atrioventricular valves are open during relaxation or diastole to allow blood to flow from the atria to fill the ventricles. The semilunar valves, however, which are between the ventricles and the arteries that carry blood out of the heart, are closed during relaxation to prevent regurgitation of blood that has just been pumped out of the heart back into the heart. So when the heart is relaxing and filling, it is filling with blood that is returning to the heart via the atria. The valves then open and close in reverse during contraction. During contraction, the atrioventricular valves are closed due to the increased pressure of the ventricles as they get ready to contract. That closure of the atrioventricular valves prevents blood from regurgitating from the ventricles back into the atria. The same pressure that closes the atrioventricular valves also opens the semilunar valves to allow blood to leave the ventricles into the large arteries. So it's a low pressure during relaxation or diastole that opens the atrioventricular valves and closes the semilunar valves, and it is the high pressure during systole or contraction that closes the atrioventricular valves and opens the semilunar valves. So let's look at this again with respect to the opening and closing of the atrioventricular valves. We have the tricuspid on the right and the bicuspid or mitral on the left. When open, the rounded ends of the cusps project inferiorly into the ventricular chamber, and this is because the papillary muscles in the ventricles are relaxed and the chordae tendineae are slack. The low pressure in the ventricles during relaxation allow these valves to open, and blood can then move from the higher pressure in the atria down into the ventricles. When the ventricles begin to contract, the increased pressure within the ventricles puts pressure on the blood and drives the cusps of the tricuspid and bicuspid valves upward until the edges meet and the valves close. This is accompanied by contraction of the papillary muscles that tighten the chordae tendineae and prevent the valve cusps from opening up into the atria chambers, thereby preventing backflow or regurgitation of blood from the ventricles into the atria. So here we have a depiction of how the cusps of the bicuspid valve relax and open during diastole or relaxation of the ventricles. And you can see that the chordae tendineae are slack. However, during contraction of the ventricles, the cusps of the bicuspid valve move upward and close, and the contracted papillary muscles then pull on the chordae tendineae and keep those heartstrings taut and help to keep the cusps of the valves closed. Let's look at those semilunar valves. The semilunar valves separate the ventricles from the large arteries that take blood out of the heart. On the right, we have the pulmonary semilunar valve that separates the right ventricle from the pulmonary trunk. And on the left, we have the aortic or left semilunar valve that separates the left ventricle from the aorta. Each is made up of 3 moon-shaped cusps that prevent backflow from the arteries into the ventricles. The semilunar valves are open when the pressure in the ventricles exceeds the pressure in the arteries, and this occurs as the ventricles contract or during systole. The semilunar valves close when the ventricles relax and the pressure is lower than the pressure in the large arteries, and the slight backflow of blood into the cusps closes the semilunar valves and prevents regurgitation or backflow of blood into the ventricles. Note that there are no similar valves between the veins and the atria receiving blood returning to the heart, as atrial contraction compresses those openings so that blood doesn't usually flow from the atria back into the veins as the atria contract to move the blood into the ventricles. So in these superior views of the heart, we can see that the semilunar valves open and close opposite to the atrioventricular valves. So in diagram D on the top left of our page, the pulmonary valves are closed and the atrioventricular valves, the tricuspid here, and bicuspid are open. So this heart is in the diastole or relaxation phase. On the right, in diagram E, we have a superior view of the heart with the atrium removed so that we can see these valves. Here, the atrioventricular valves are closed and the semilunar valves are open. So this represents the contraction or systole phase of the heart cycle. Also shown below in the cadaver views. The anatomy of the heart and its associated blood vessels create distinct pathways for blood flow or circulations. The blood flows with each heartbeat in 2 separate closed circuits so that oxygen-rich blood does not mix with oxygen-poor blood or deoxygenated blood. However, these 2 circulations are moving blood at the same time as well as in series so that the blood flow out of one circuit becomes the input of the other. The pulmonary circulation is created by the blood flowing through the right side of the heart from the right atrium down to the right ventricle, which is then pumped up into the pulmonary trunk and arteries to the lungs. On the left side, we have blood returning from the lungs into the left atrium, down into the left ventricle, and then pumped out into the aorta and its branches to form the systemic circulation that provides blood to the other tissues of the body. Part of the systemic circulation is the coronary circulation, and this is created by the blood vessels that bring blood deep into the muscle tissue itself. And the heart requires its own circulation, just like other organs, because the blood passing through the chambers does not have sufficient time to diffuse into all of the muscle cells that make up that thick myocardium. So blood that is pumped out from the left ventricle enters a coronary circulation that brings blood into the muscles of the heart. That blood, once it passes through the muscle tissue, then exits and drains into the coronary sinus that brings that deoxygenated blood into the right atrium. So here's an overview of the circulations working in series. Shown in blue is the deoxygenated blood coming from the systemic tissues as well as the heart muscle, returning to the right atrium, which passes by the tricuspid valve into the right ventricle and is pumped from the right ventricle past the pulmonary valve into the pulmonary trunk and pulmonary arteries that bring the blood to the left and right lungs. In the lungs, gas exchange occurs so that carbon dioxide is removed and oxygen is loaded into the blood. This oxygen-rich blood then returns to the heart via the pulmonary veins into the left atrium, which moves the blood past the bicuspid valve into the left ventricle. The left ventricle pumps the blood past the aortic valve into the aorta and the systemic arteries, which brings blood to the tissues of the body as well as the heart. So it's closed circuit, divided into two distinct pathways. Again, the output of one becomes the input of the other. Also note that while we've drawn it as a continuous circuit, the two sides of the heart pump simultaneously so that the same volume of blood is moving from the right side of the heart to the lungs as is being ejected from the left side of the heart to the body systemically with each heartbeat. Again, the pulmonary circulation involves the right side of the heart that brings oxygen poor or deoxygenated blood from the body to the lungs, where the blood unloads carbon dioxide and picks up oxygen. The pulmonary veins then carry the blood back to the left atrium. The systemic circulation is created by the left side of the heart as its pump, where oxygen-rich blood returning from the lungs is delivered to all the body cells. Oxygen is released in the tissues, and carbon dioxide created by cellular metabolism is picked up, and then that blood returns to the right side of the heart via the systemic veins and the vena cava. Here we have an internal view of the heart and the associated blood vessels that show the path of blood flow through the heart. So again, blood returns to the right atrium via the two vena cava and the coronary sinus. That blood then moves down into the right ventricle, up past the pulmonary valve into the pulmonary trunk, number 3, into the right and left pulmonary arteries that eventually branch into the pulmonary capillaries that allow for gas exchange in the lungs, where carbon dioxide is unloaded and oxygen is loaded into the blood. Number 5, the blood returns to the left atrium via the pulmonary veins. Blood from the left atrium, number 6, moves down into the left ventricle, number 7, up past the aortic valve into the aorta, number 8. So structures on the right make up the pulmonary circulation, and the structures on the left make up the systemic circulation. We mentioned that the heart has its own blood supply that makes up the coronary circulation, and it is formed by two arteries that branch from the ascending aorta just as the blood leaves the left ventricle. So branching off the aorta, we have the right and left coronary arteries, and they supply oxygen-rich blood to the myocardium of the heart. The branches of the right and left coronary arteries extend via the sulci to both atria and ventricles on either side of the heart. The coronary circulation is unique in that there are several connections where two or more arteries supply blood to the same region and provide alternate routes in case either branch of an artery is obstructed. So these connecting pathways are called anastomoses, and they provide for collateral circulation. Blood is allowed to enter the coronary arteries mainly during the relaxation phase of the contraction cycle. There are two reasons contributing to this. Number one, during systole or contraction, the flaps of the aortic valve are open up into the aorta and partially block the opening to the coronary arteries. It is only during relaxation, when the aortic semilunar valve is closed, that these openings are patent, allowing blood that is in the aorta to then flow into the coronary arteries. Secondly, during contraction, the contraction of the heart actually compresses the coronary arteries and its branches that supply blood to the myocardium. And that compression limits blood flow through these coronary arteries. So it's only during diastole that the coronary arteries are open and allow blood to flow and supply the myocardium. Within the myocardium, the arteries branch into the coronary capillaries, where the gas exchange occurs similar to what occurs in the systemic circulation. And then the coronary veins collect that deoxygenated blood from the heart tissue and drain it into the coronary sinus that runs on the posterior aspect of the heart and empties into the right atrium. Nutrients to the heart muscle, as well as oxygen, are provided by the coronary arteries that make up the coronary or cardiac circulation. The coronary arteries branch from the ascending aorta and encircle the heart and branch deep into the myocardium. The right coronary artery supplies the right atrium and the superior right ventricle. Note that the right atrium contains the pacemaker of the heart, known as a sinoatrial node, so blood flow through the right coronary artery is significant for supplying the tissue that leads to heart contraction. The major branches of the right coronary artery are the marginal branch and the posterior interventricular branch. The marginal artery and its tributaries supply the muscle of the anterior right ventricle and some of the lateral aspects, while the posterior interventricular branch supplies the lateral and posterior right ventricle, as well as the posterior left ventricle and the posterior interventricular septum between the two ventricles. The left coronary artery branches to supply the tissue of the left atrium, as well as parts of the left ventricle. The major branches of the left coronary artery are the anterior interventricular branch, also known as the left anterior descending artery, or the LAD, and the circumflex artery. The left anterior descending or anterior interventricular branch supplies the muscle of the right and left ventricles, as well as the anterior interventricular septum, as well as the apex of the heart. The circumflex branch supplies the lateral and posterior left ventricle, as well as parts of the left atrium. Once blood passes via these arteries into the capillaries that supply the muscle cells of the heart and gas exchange occurs, the blood then leaves the tissues and enters the cardiac veins, which collectively drain into the large coronary sinus that brings the oxygenated blood from the heart into the right atrium. All right, we will now move into some histology and consider cardiac muscle tissue. Cardiac muscle cells, or myocytes, are called fibers because they're somewhat branched and elongated. Unlike skeletal muscle, cardiac muscle fibers are connected end to end and interlock with each other and are held in place by intercalated discs that contain proteins called desmosomes that provide strength and hold the cells together. Connecting the cells are passageways called gap junctions, and these are significant and somewhat unique to cardiac muscle cells in that they allow muscle action potentials to pass quickly from one muscle cell to its neighboring cell, and the signals pass quickly through the myocytes of each chamber so that the chambers contract as a unit. There are specialized muscle cells called autorhythmic fibers, and these form the cardiac conduction system or pathway. The cells that are autorhythmic can spontaneously depolarize because they have slow leak sodium channels that allows sodium to diffuse slowly into these cells, depolarizing them and bringing them to threshold. Each set of fibers depolarize at their own rate. However, the rate of depolarization from the autorhythmic fibers in the right atrium that make up the sinoatrial node is the fastest, so that it sets the rate for depolarization for the rest of the cells. The heart muscle tissue is made up of contractile fibers, these myocytes or muscle cells, that create the powerful contractions that propel blood from one chamber to the next and out of the ventricles into the large arteries. So here we have a picture showing the branched cardiac myocytes and how they are interlocked and connected by desmosomes. And here we also see the gap junctions, which are the passageways between adjacent cells that allow for muscle action potentials to pass quickly from one cell to the next so that the chambers contract as a unit. The heart contracts rhythmically and autonomously due to the existence of autorhythmic cells that make up the cardiac conduction system. The cardiac conduction system begins with the autorhythmic cells of the sinoatrial or SA node, which acts as a pacemaker for the rhythm of electrical excitation in the heart. The cells of the sinoatrial node depolarize regularly at a rate faster than the other autorhythmic cells, so that its rate of depolarization overrides that of the other cells and sets the rhythm of heart contraction and relaxation. The conduction system provides a pathway for signals that originate in the sinoatrial node to spread through the heart, leading to a regular and coordinated rhythm of contraction. So let's look at the pathway for the progression of the signals through the heart. The signals originate with the spontaneous depolarization in the sinoatrial node, which is located in the right atrial wall. The depolarization wave or the action potential will then pass quickly through the cells of the atria via those gap junctions, causing the cells of the atria to contract together. The signal will then pass down to the atrioventricular node, a bundle of cells located in the superior interventricular septum. The signal is partially delayed at the AV node before continuing to the atrioventricular bundle, also known as the bundle of His, also located in the interventricular septum. From here, the electrical signal or the action potential wave will pass into the right and left bundle branches, which are located in the interventricular septum, and move towards the apex of the heart. From the apex of the heart, the signal will then pass into the Purkinje fibers, which branch from the apex upward into the myocardium of the ventricles. The Purkinje fibers then bring the signals to the muscles of the ventricular myocardium, causing the ventricles to contract. So this pathway ensures that the atria contract together, followed by contraction of the ventricles, and this ensures coordinated contraction and maximal filling and then emptying of the ventricles, leading to blood ejection. So here we have the cardiac conduction system outlined for us. It begins at the sinoatrial node in the right atrium, the spontaneous depolarization of the autorhythmic cells of the SA node. The electrical signal generated there then passes through the atria, leading to atrial contraction. That atrial contraction occurs while the ventricles are still relaxed and will help to remove any blood that's still left in the atria down into the ventricles to complete the filling of the ventricles during the relaxation phase known as diastole. The signal then passes to the atrioventricular or AV node located in the superior interventricular septum, and then down to the atrioventricular bundle or bundle of His. From there, the signal will pass via the right and left bundle branches located in the interventricular septum to the apex of the heart, and then to the Purkinje fibers that branch into the myocardium of the ventricles. These Purkinje fibers bring the signals to the ventricular myocytes, causing them to contract as a unit. So this pathway ensures that the atria contract first to complete the filling of the ventricles, and then eventually the signal passes to the Purkinje fibers, which cause the ventricles to contract. Contraction of the ventricles closes the AV valves and opens up the semilunar valves, leading to ejection of the blood out of the ventricles into the large arteries during systole. Let's look at the contraction of the cardiac myocytes or contractile fibers. The mechanism of cardiac cell contraction is similar to skeletal muscles. So there's a similar arrangement of the myofibrils, the myofilaments, the actin and myosin, which overlap to form sarcomeres. The actin is still guarded by tropomyosin and troponin and requires calcium for interaction and contraction to occur. Now, action potentials that originate in the sinoatrial node eventually pass to the myocytes of the atria and the ventricles, where they trigger opening of voltage-gated sodium channels that allow a sodium influx that causes a sharp depolarization. When the membrane is depolarized, this opens calcium channels in the sarcolemma of these muscle cells, and this is unique to cardiac myocytes. Calcium is then going to flow into the cells and cause a sustained depolarization, because at this time, some potassium channels also open and allow for the outflow of potassium. So the influx of the calcium is matched by the outflow of the potassium, and that creates a sustained depolarization or the plateau phase. This also creates a sustained contraction of the myocytes, which allows them to eject the maximum amount of blood from the ventricles. Once the calcium channels close, more potassium channels open, and the outflow of potassium leads to repolarization and eventually the restoration of the resting membrane potential. Note that the calcium that flows from the extracellular space into the cell not only contributes to the action potential, but also allows for myosin and actin to interact by binding to troponin, which leads to the moving or shifting of tropomyosin so that the contractile proteins can interact. Therefore, the influx of calcium that's part of excitation is coupled to the contraction interaction itself. So we call this excitation-contraction coupling. Therefore, anything affecting the calcium movement influences the strength of cardiac muscle contractions. Cardiac myocytes have a long refractory period or relaxation phase that allows the chambers to refill with blood. So when the cells are repolarizing and are refractory, they are in the relaxed phase, and this is also the filling phase for the chambers of the heart. ATP that powers contraction and allows for restoration of the resting membrane potential in these cardiac myocytes is mainly formed from aerobic cellular respiration. The heart cells have the capacity to use fatty acids and then glucose as their main fuel sources, but cardiac myocytes can also make ATP from lactic acid, amino acids, and ketone bodies. Muscle cells also have the capacity to make ATP quickly from creatine phosphate, which is catalyzed by the enzyme creatine kinase. On this diagram, we have a depiction of the membrane potential as it changes in the pacemaker cells of the autorhythmic sinoatrial node cells. Note the slow depolarization and due to the slow leak of sodium into these cells. As threshold is reached, voltage-gated sodium channels open, and you get that sharp depolarization followed by repolarization. And the action potentials that are generated in these pacemaker cells then spread via the gap junctions to the cells of the atria and down the conduction pathway and via the Purkinje fibers to the cells of the ventricles, leading to opening of their sodium voltage-gated channels and then their depolarization and repolarization or action potentials. The previous slide showed us the action potential profile of the autorhythmic cells of the sinoatrial node created by the slow influx of sodium through leaky channels. Those do not exist in the myocytes of the atria and ventricles, which rely on the signals that originate from the sinoatrial node to pass to these cells via the conduction pathway. So in this slide, we have the action potential in a ventricular cardiac myocyte or a ventricular contractile fiber. The signals that arrive from the sinoatrial node cause the opening of voltage-gated sodium channels in these ventricular myocytes that lead to a rapid influx of sodium and a sharp or rapid depolarization. When depolarized, the sarcolemma opens its calcium channels as well as potassium channels. And the influx of calcium is balanced by the outflow of potassium. So this results in a sustained depolarization. And note that while the cell is depolarized, it is also contracting so that cardiac myocytes don't twitch like skeletal muscles. They experience sustained contractions, and this maximizes the amount of blood that is ejected out of the heart as the ventricles contract. Eventually, the calcium channels close and the potassium channels remain open, and this results in repolarization as the potassium flows out of the cells. Note that while the membrane is depolarized and is in early repolarization, it is in a refractory period. Again, this is when the cells cannot respond to new action potentials. And the refractory period in muscle cells of the heart is much longer than that in the skeletal muscles and lasts longer than the contraction itself. As a result, another contraction cannot begin until relaxation is well underway.